grabación, entonces ahora sí.
Vale. Ahora ya hay que cuidar lo que se dice. Sí, no problema.
Okay.
Welcome everyone. About to start the talk. So please, for the respect of the doctor, be in silence. Welcome. The talk is going to be about metabolic switches and epigenetic marks in the blueprint of dynamic gene regulation by Dr. Lorena Aguilar. Dr. Lorena Aguilar is originally from Spain, earned her bachelor degree in biochemistry from the University of Navarra in 2002. She conducted a research internship at the Spanish National Cancer Research Center before obtaining her PhD in biomedicine from the Institute for Biomedical Research of Barcelona in 2009. The same year, she received a prestigious MBO postdoctoral fellowship to conduct research at the University of California. During her 6-year postdoctoral training, she explored the molecular mechanism linking cellular metabolism with the circadian clock. Her groundbreaking research demonstrated how the circadian clock machinery shapes the three-dimensional genome structure to regulate rhythmic gene expression with findings published in Nature Structural and Molecular Biology. In 2016, she joined the Institute of Biomedical Research at UNAM, where she has since published 20 research articles, secured national and international funding, supervised multiple students, and taught over 30 postgraduate courses. She currently serves as a president of the Academic College of the same institute and is a Level 2 member of the National System of Researchers. Welcome, Dr. Aguilar, with a big round of applause.
Oh. Thank you very much for the presentation. Also, thank you very much to the organizers for inviting me to give this talk here. I'm very happy of being here and for hosting me. I'm going to talk to you about what is circadian rhythms and what we do in the lab, what we are doing as research in the lab. So there are activities, behaviors, or physiological processes that in nature appear cyclically in many living forms. They allow us to adapt to recurring changes in our environment. And you can see these behaviors in living organisms such as plants, or in the migration of the birds, or in our pets. And the interesting part is that these cycles are genetically encoded and they are endogenous. So there is a cycle that is very persistent since millions of years ago. That is the cycle that Earth makes in the rotation of its own axis, making these day-night cycles that we experience every 24 hours. And thanks to this, there is the evolution of circadian rhythms that are 24 hours period cycles in biochemical, physiological, and behavioral functions in most living organisms, from bacteria, fungi, plants, and animals. Every living organism has circadian rhythms, these cycles that are endogenous. They are genetically encoded, and they must, for their efficiency, they must synchronize in a daily basis with the environment through specific signals. In mammals, the most important signal that synchronize circadian rhythms is light. No? These signals are called zeitgebers. Everybody can relate their own circadian rhythms. Probably the most evident is the sleep-wake cycle, but there are a lot of other circadian rhythms in our daily performance. We can find, for example, at the behavioral level, there are times where it's easier to, sorry about this, it's easier to be alert, no? The coordination is better. Also, there are another circadian rhythms in physiology. For example, you can experience blood pressure rhythms around the day, but also body temperature appears in a circadian basis. There are times of higher body temperatures and times of lower body temperature around the day that is persistent every other day. And also there are rhythms in biochemical parameters, such as, for example, many hormones appear cyclically in the blood. For example, melatonin secretion is circadian. You can probably melatonin as the hormone for sleep, but there are many other hormones that are circadian. For example, testosterone and many other sexual hormones, but also all those hormones that control glucose homeostasis, such as insulin, glucagon, appear rhythmic in circulation. But also a lot of metabolites, such as many lipid species, or for example, cholesterol and many others appear with in a circadian fashion in the circulation. Okay. Circadian rhythms synchronize behavior and physiology with environmental changes. They serve to anticipate recurring events such as like dark cycles or feeding times, and they regulate daily activity and rest patterns in living organisms around the day. The most important time giver in mammals is light. This light is the signal that an organism, a mammal, perceives and uses to synchronize its physiology and behavior with the environment. And not only the presence or absence of light is the reason that synchronizes circadian rhythms, but also the wavelength or the color of the light can have an impact on how circadian rhythms appear in our physiology and organism. How is it possible? Well, I will explain in this little video how this schematic ahí abajo. So every morning we wake up and we ideally expose ourselves to the sunlight, no? And then sunlight goes into the eye, into the retina, and besides collaborating to make visual images possible, it also allows to impact some special cells in the base of the retina that project through the retinohypothalamic tract to a non-photic area in the base of the brain, in the hypothalamus, that is called the suprachiasmatic nucleus. This area contains the mammalian circadian clock, the mammalian central clock, and it's very interesting because it has a very important kind of neurons that have the particularity that they are unique. This is a unique particularity that their activity oscillates in synchrony. So these neurons fire together. It's a network, and its firing is about 24 hours period. No, it's a network that is synchronized through the light from the retina. So this suprachiasmatic nucleus that encodes the internal pacemaker for circadian rhythms anatomically is located in the third ventricle in the brain, in the hypothalamus in the brain, and the firing of their neurons is oscillating in synchrony. Here you have a video of a suprachiasmatic nucleus in a mouse brain that is fluorescently labeled, and if you can play this video also, it's a time lapse, and you can see that the fluorescence is oscillating in synchrony in all the cells in this area. And this is a particularly exclusive from the suprachiasmatic nucleus. What light does is synchronizing this firing, no? For example, synchronizing this firing. So the light goes into the retina. There is this ganglion cells in the base of the retina that goes through the retinohypothalamic tract directly to the suprachiasmatic nucleus, synchronize these neurons, and they start to fire with 24 hours period. And this, through neural innervation, it can relate this information to other nucleus or areas in the brain, in the same hypothalamus and in other areas in the brain, generating these rhythms in physiology and behavior and biochemical rhythms. Additionally, clocks are everywhere. So even through the suprachiasmatic nucleus is the circadian pacemaker that can integrate the external signaling. After the integration of this signaling through the suprachiasmatic nucleus, neuronal signaling and hormonal signaling relates this information to other clocks that are everywhere in every tissue in the body. So the perfect synchrony amongst all of these oscillators give rise to this sleep-wake cycle, thermoregulation, food intake, blood pressure, and metabolic rhythms, and also to these rhythms in physiologically active hormones such as melatonin or cortisol, right? And then not only, it's important to understand or to keep in mind that not only light can synchronize circadian rhythms, but also there are other time givers or state givers that can synchronize directly these peripheral clocks to reinforce this synchrony amongst the different tissues, such as, for example, food, temperature, activity, physical activity, or social say givers, like the way we relate to each other or with the screens that emit blue light at night, for example. So the perfect synchrony keeps time, but if we disrupt circadian rhythms, it can lead to impaired metabolism. For example, how can circadian disruption happen? For example, through the exposure to artificial light at night, screens, telephones, computers, TVs can disrupt circadian rhythms because lights come at an hour that ideally should not be there. Late night eating also can disrupt circadian rhythms. Night shift works can lead to circadian disruption or social jet lags, such as, for example, creating a jet lag during the weekends where the schedule or the normal schedule is not maintained. It can lead to circadian disruption, and this gives rise to several, it has been shown that it's associated with the appearance of many diseases such as cardiovascular diseases, endocrine and metabolic problems, for example, also cancer or other cognitive related diseases. There is also genetic evidence of the relationship between circadian rhythms and metabolism, such as, for example, those that arise from mutant mice. So we can have some mice that can be transgenic for some of these factors that encode endogenous circadian rhythm genetically. For example, the clock mutant mice, that is a mutant that do not show circadian rhythms due to a mutation that they have in their genome. What is evident in these mice is that they develop hyperlipidemia, increases susceptibility to obesogenic diet, so they get fat very easily. They have age-associated hyperglycemia, hypoinsulinemia in young mice, and another kind of metabolic syndromes that come from the studies in these genetically modified mice. So circadian rhythms and metabolism are very, very linked between each other. So in 2007, this field exploded because the Nobel Prize in Physiology or Medicine was given to these three outstanding researchers, jointly to Jeffrey Hall, Michael Rovach, and Michael Young, for their discoveries of molecular mechanisms controlling the circadian rhythm. And what are those mechanisms? Well, in my lab, these mechanisms, the molecular mechanisms which control circadian rhythms are the focus of attention, because they are mostly transcription factors that regulate the expression of genes. So transcription factors, these are proteins that bind DNA. They are recruited to the DNA, to specific sequences that are recognized by these transcription factors, and their binding can lead to transcription. In this video, I'll show you how this molecular clock works. Basically, what happens is that there are these transcription factors in these two boxes, like the green and blue box. It's started. No. It's started the video, right? Yeah, no. So these transcription factors that are the molecular machinery are called bimalin clock. They come together to the genome, to a very specific sequence in the genome that is called EBOX. It's a regulatory element that controls the expression of a number of many genes. But amongst those genes that are controlled by bimalin clock are also the negative regulators of transcription, that are the transcription factors period and cryptochrome. So there are 3 period genes and 2 cryptochrome genes in the mammalian genome. And when bimalin clock transcribes them, they are transcribed into mRNA and they are translated into protein in the cytoplasm. In the cytoplasm, the bimalin clock proteins are come together, generating the transcriptional repressor complex that is called the period complex, and it translocates back into the nucleus. And when it reaches adequate concentrations, what happens is that it can bind to the activator and recruit it and sequester the activator and remove it from the DNA. In a way, the transcription is stopped, and then in a mechanism, depending of the gradation of this negative regulator, the negative regulator is cleared from the nucleus, and the activator is free again to join again to the DNA, bind there, and start another cycle of transcription. The interesting part of this cycle, of this autoregulatory cycle of transcription and translation, is that it takes to happen about 24 hours, in a way that every 24 hours this happens and gives rise to circadian rhythms in transcription of the genes. So in these maps, you can see the expression of many genes in the liver or in the muscle of mice that have been sacrificed every 4 hours. No, these are two days, no, of the recording of the expression of the genes. Every horizontal line is a gene, and then in green there is a low expression of the gene. In red there is high expression of the gene. So every gene that is in this map, there are thousands of genes. 4,000 more or less in the liver, is one fourth of the transcripts from the liver. Appears and goes in a circadian fashion. Every 24 hours, it's transcribed and it's shut down. Also in the muscle, there are a lot of genes that follow a circadian fashion and are regulated by this molecular mechanism. And these genes are very, very important for the physiology of these tissues. Every tissue has its circadian transcriptome. So from a circadian transcriptome, sometimes eventually these transcripts are translated into proteins. It comes a circadian proteome. There are a lot of proteins that appear in the cell in a circadian fashion. They appear and disappear every 24 hours. And then what is interesting also is that many of these proteins are also enzymatic regulators of many metabolites biosynthetic pathways. No, for example, they are rate-limiting enzymes that can control the pace that metabolic pathway flows, generating a circadian metabolome that can be investigated through techniques such as mass spec. No? So yeah, there are circadian transcripts, circadian proteins, circadian metabolites. But the question that we try to investigate in our lab is how are transcriptional rhythms generated? No? Because it's not only that these proteins bind and evict every 24 hours from the chromatin or from the genome, but also rhythms or changes, these fluctuations in transcription require high plasticity on the DNA, on the chromatin. And the mechanisms that are oriented to impose this plasticity on the DNA are called epigenetic mechanisms. And so this epigenetic control cooperates with the molecular clock machinery to generate these 24-hour rhythms. And this is the focus of research in my lab. Epigenetics is the study of heritable changes in gene function that do not involve alterations to the underlying DNA sequence. And epigenetic changes can be influenced by environmental factors, lifestyle, and experiences, and they play a crucial role in regulating gene activity. So some epigenetic mechanisms are illustrated here in a very simple way, but well, they consist of mainly of covalent, for example, on covalent modifications of the DNA, no, such as these DNA modifications. These red dots represent a covalent modification. Generally, it's a methylation of a cytosine that modifies directly the double helix of DNA. But the DNA in the cell is not naked. Is on the contrary-wise, is tightly linked to proteins, and half of these proteins are histones. Histones are basic proteins, are the most basic proteins in the proteome, and they associate to each other in a way that 8 histone proteins generates a nucleosome. This is that are here illustrated at these barrels. This nucleosome consists of 2 turns of DNA helix, 184 base pairs that organize around these 8 histone proteins. And these histone proteins are also subject of modifications through the introduction of small chemical groups such as acetylation or methylation that are the epigenetic marks. No? And these epigenetic marks that are here illustrated with these dots contribute to allow the function of the genome. For example, silenced genes or activate genes. So they are very, very, very involved in the way that the DNA is transcribed, right? Also, there are other higher order chromatin organization and the nuclear positioning that influences the way that transcription is made in every moment in the cell. Oops, yeah. Okay. So here it comes a little bit more difficult. So let's see. So this is, for example, an experiment. This is to illustrate how the circadian machinery contributes to cooperates with the epigenetic modifications to give transcriptional rhythms in the genome. So, sorry. So this is an experiment that is called CHIP. chromatin immunoprecipitation of this protein that is BMAL. And these samples from mouse liver are taken every 4 hours. No? The CT0 is the time that light is open, is on in the vivarium, and CT12 is the time that light is off and is shut down in the vivarium. So this is taken, these samples are taken every 4 hours, and then these tracks here represents the amount of BMAL protein that is located at this position of the genome. This is a gene that is expressed in a circadian fashion. It oscillates in a circadian manner, its transcription. So BMAL comes to the genome at this time, no, during the light period, and it's evicted from the genome, from the chromatin at this time, no? And this is what I show you in the little video of the work that of circadian machinery. But if we study the epigenetic marks in this genome, in this position also, what you can see is, for example, that remethylation of lysine 4 of histone 3 also comes into the genome in a circadian fashion, and acetylation of lysine 9 of histone 3 also is circadian. There are times of the day that comes together with BMAL, and these epigenetic marks contribute to create an epigenetic environment that is permissive to transcription, imposing rhythms of transcription into this gene at certain times of the day. So also, the molecular clock can control the pace at which many metabolic pathways flow, and this is an example. This is the salvage pathway of the NAD, the nucleotide of nicotinamide and adenine. This is a very important metabolite that is in the body and controls, for example, oxidative reactions. No, it's a very important metabolite, and here there is the pathway that leads to the synthesis of this metabolite. And the amount of this metabolite that is in our cells is mostly controlled by this recycling pathway. It's a circular pathway that comes from, it's called the NAD salvage pathway, that recycles NAD from nicotinamide, that is the bypass metabolite of the action of these NAD consuming enzymes, that there are several NAD consuming enzymes. So the pace at which this pathway flows dictates the amount of this metabolite inside of the cell. And the pace at which this pathway flows is controlled by this rate limiting step that is catalyzed by this enzyme that's called NAMPT. So when NAMPT is here and is working, it flows, and then there is a rise in NAD. When NAMPT is not here, this pathway is stopped here at the level of nicotinamide, and there is a drop in NAD levels. No? The interesting part is that NAMPT is one of those genes that is rhythmically expressed through the binding of the molecular clock machinery into the regulatory elements of the NAMPT gene. So NAMPT is expressed in a rhythmic fashion from its transcripts, is traduced to the protein, to this enzyme, and it makes this pathway oscillate and flow in a circadian manner in a way that NAD appears rhythmic in our cells. So during my postdoc in California, in the University of California, we describe this tight collaboration between the circadian epigenome and the cellular metabolism in a way that when these molecular activators of the clock are bound to chromatin, they cooperate with an enzyme that is called MLL1 that promotes the introduction of these chemical groups, the methylation of lysine 4 of histone 3, that is an epigenetic activating mark. So the convergence of this methylation and acetylation that is elicited by the clock machinery in this region of, it might be a promoter of a gene, creates an epigenetic environment that is permissive to transcription, leading to transcriptional activation of many genes in the transcriptome. Among those genes, there is the transcription of NAMPT, that is the enzyme that I showed you in the slide before, and is translated into the protein, and then the metabolic pathway flows and NAD levels rise. NAD levels rise, and then it activates a protein that its activity depends on NAD. It's called SIRT1, leading to its activation. SIRT1 is a deacetylase, and it starts a deacetylation cycle. It's active, and then it starts a deacetylation cycle over the clock machinery proteins that are acetylated, leading then to inactivation and eviction from chromatin. And this creates an epigenetic environment that has another that do not show these epigenetically active epigenetic marks. They are marks, the marks that are here are not transcriptional activators anymore. And then this leads to transcriptional repression, showing or illustrating how cellular metabolites contribute with the circadian machinery and the epigenome to generate these cycles in expression and repression of many genes in the genome. Well, by that time, also when we described this mechanism, there was a lot of research showing how this metabolite NAD can be an efficient drug to treat many pathological situations related to metabolic dysfunction, such as, for example, diet and age-induced diabetes, or, for example, mitochondrial dysfunction, or high-fat diet-induced obesity, diabetes and diabetic neuropathy in mice. It could be recovered by raising endogenous NAD levels, either pharmacologically or genetically, or through diet. You can reverse many of these pathological situations, and NAD is a potential therapeutic target for treating this metabolic dysfunction. But none of these papers none of these research, there were hundreds, took into consideration that this metabolism, in fact, normally appears with a circadian fashion. Then we wonder if this was important. The circadian rhythms of NAD, endogenous NAD levels in a normal situation, was important for the effects of NAD as a therapeutic agent to treat the pathophysiology of obesity and metabolic disruption. So to do so, we created with the outstanding PhD student and postdoc Quetzal and Lucia, we created this mouse model of diet-induced obesity, and we had these groups: a group of mouse model that was normal control mice, and these three other groups of obese mice that have been fed a high-fat diet, and they showed marks of diabetes in mice, these diabetic mice. And then we raised the NAD levels at 2 different times of the day. One, that is CT11, CT12, is just before the mice wake up, no? It's at the onset of the rest phase. And these ones at CT23, CT24 was just before the end of the active phase, yeah? And then here you can see how NAD levels are measured by HPLC. You can see that in normal mice, it's in black, controlled diet. It's in black, the NAD levels are endogenously and they are rhythmic. In the high-fat diet-fed mice, in this group, NAD levels are flat, they do not oscillate anymore. And this is because obesity leads to circadian disruption, so they cannot oscillate anymore. And in these treated mice, the ones that are treated at CT11, CT12, you can see the rise of NAD levels at the same time that normally in control mice are oscillating, but these are still obese mice. And in these mice that have been treated at CT23, CT24, the rhythms are inverted, right? You can see the rise 12 hours later, no? These are inverted rhythms, but it also rises, but at a time that normally it does not happen. So then we thought, oh, okay, let's evaluate if the treatment works the same or similar independently of the time of the day that you apply it. No, this is important, for example, for therapeutics in case that we need to translate this treatment to the clinic, right? Then what we saw, well, here you have the mice in green that have been treated at the same time that normal circadian rhythms of this metabolic happen. So the ones that are treated before the mice wake up. So you can see at the beginning of the treatment how they lose weight. No, they start losing weight. It's the first week. The second week they keep losing weight and they stabilize. And normally they should keep. They are still losing weight at the end of the treatment. But the other mice that are treated at the 12 hours later, the first week appear to lose a little bit of weight, but not much, and they start to wane. So the treatment is not efficient, at least for losing weight. No, this is a very efficient drug for losing weight. And this is not dependent on the food intake. They eat the same, and the change in total body weight is only significant in the mice that have been treated at the time that mimics the normal situation of NAD levels in the control mice. And we did a lot of measurements of metabolism, and we'll reach the conclusion that the drug was, the efficiency of the drug was best when it was administered right before the mice were going to wake up. It was not working when it was administered right before the mice were going to sleep, right? Then we wonder what was going on, why this happened, because from my previous experience, NAD has an impact on the expression of the genes. And then we say, oh, let's see which genes have been recovered, their expression, after the administration of the NAD. And then we did this experiment at 2 times of the day, at day or at night, right? This is the transcriptome of the liver during the day or during the night. We saw that after the treatment in the mice, in the treated mice, there were a number of genes that were recovered. Their expression was recovered to the levels of the control mice. No, so the metabolic, this drug was recovering a set of genes, their expression at day, and also another set of genes it was recovering their expression at night. No, and then when we tried to investigate which genes were these, we found that there were a lot of biological processes that were restored after the administration of the drug. So this drug during the night time, that is because mice are nocturnal, this is the time that mice are awake. We saw that there were a lot of metabolic processes, such as lipid metabolic processes or glycerolipid or organoid acid metabolic processes or lipid storage related to lipid metabolism that were restored to control conditions. So the drug was controlling very well the homeostasis of lipid metabolism during the night. That is when the mice are eating. During the day, that is when the mice are sleeping, we saw that the drug was restoring a lot of pathways related to the immune system. No, it was restoring pathways related to regulation of the immune system, innate immune system, defense response to other organs. So the inflammatory mark that is concurrent with obesity was erased by the treatment during the day when mice were resting. So the inflammation in these mice had disappeared. We further explored some pathways. Some of them we start to explore because if the drug was differentially working day or at night, we thought that we must study the circadian machinery to understand what is doing this drug to the circadian machinery. And then we found a very striking result that was that this drug is a very potent synchronizer of the circadian machinery in the cells. As you can see here, well, this is a Western blot of circadian proteins. So these are the mice that are treated at PD12, so just before the mice are going to wake up. And these are the mice that are treated at CT24, that is just right before the mice are going to sleep. So if you see, for example, here, this is a circadian protein, Rever alpha, you see that it appears at these 2 hours, no, well, between day and night. But in the mice that were treated 12 hours later, the protein is present in the opposite way at 2 different hours that normally should not be there. Also with period, cryptochrome, so all the rhythms of the circadian machinery were inverted in these mice. At the protein level, but also at the transcriptional level, is very remarkable how this NAD can completely invert, here in blue you can see, the rhythms of expression of these circadian proteins, but also of the genes that are controlled by the circadian rhythms. And it was doing so without disrupting behavioral rhythms. No? So mice were sleeping at the time that they should, and they were waking up at the time that they normally are. And this is what this plot means. And this plot, so what's happening is that the drug was synchronizing the molecular clock machinery in the liver, so it was delaying 12 hours the expression of these proteins, and this created the synchrony between the endogenous rhythms or in the brain, or well, first with the environment, right? but also with the brain that is synchronized directly with the environment. And this circadian misalignment was opposing to the effects of the drug. And this research has been recently published in Nature Communications. And like as a take-home message, now that we are thinking a lot on precision medicine in a way that can allow us to treat a patient depending on its genetic background or in certain characteristics that are unique to each patient, we need to take also time into consideration and to understand the circadian rhythms of each patient to select the time at which each drug should be administered for better performance. No? And this happens with a lot of drugs or pharmacs. No? It's very, very extended in pharmacology. We also wanted to understand a little bit more on how NAD, this metabolite that is so important to maintain physiology, can impact epigenetic marks and epigenetic regulation. And then at the molecular level, the environment and epigenetic mechanisms are connected through metabolites, cofactors that epigenetic remodelers use to catalyze epigenetic modifications. For example, SIR1, no? The one that I showed you in some slides before during the introduction, is a deacetylase whose activity is tightly coupled to the hydrolysis of NAD. No? To do its activity, that is the acetylation of substrates, it must hydrolyze NAD to nicotinamide and perribose. No? So the presence of NAD, the Michaelis constant of SIR1, is in the range of the physiological changes of NAD in the cell. No? So high NAD levels can activate SIR1, low NAD levels will shut down SIR1. And it has been shown that activation of SIR1 is associated with neuroprotective effects, is anti-inflammatory and cardioprotective effects in the heart. It promotes fatty acid oxidation and gluconeogenesis in the liver. It diminishes lipogenesis. And in the pancreas, it can promote insulin secretion. And very importantly, in the muscle, it can lead to increased insulin sensitivity, which is very important, for example, for the treatment of type 2 diabetes. No? So with this basis, we use this model of differentiation, that is a differentiation from human mesenchymal stem cells from human donors. We can, in our lab, we can differentiate mesenchymal stem cells to adipocytes. Mesenchymal stem cells can also differentiate to osteoblasts or myoblasts through the activation of the Wnt pathway, and through the inhibition of the Wnt pathway, it can differentiate to adipocytes. No? And this is very well studied, and we know how the sequential activation of transcription factors that happens around this process of differentiation of adipocytes. No? There are a lot of transcription factors going on, and this is a sequential activation. And there are two stages: the commitment and the differentiation. The commitment to the pre-adipocytes and the differentiation to mature adipocytes. So with this, in this model, that we can reach to it in 16 days with the help of our collaborators and this remarkable PhD student, Edgar Sanchez. We established this model in the lab, and here you can see the adipocytes that are in red. This is a stain of the lipids, no? After 16 days of differentiation. Then in this model of differentiation, we treated with these drugs, NAD, and then a drug that inhibits SIR1, that is the enzyme that catalyzes the hydrolysis of NAD into nicotinamide, and a drug that inhibits the rate-limiting step in AMPT, that is FKH6. And then this drug doesn't allow this recovery of NAD from nicotinamide, and then what does is drops endogenous NAD levels. And then we treat the model with these drugs, and then what we saw is that each drug has a different effect in the adipogenic differentiation, in a way that treating with NAD or with EX527, the inhibitor of SIR1, leads to a very poor adipogenic differentiation. So cells cannot go through the adipogenic process, here in yellow and in red. However, if you promote a drop in NAD, endogenous NAD levels, with this drug, FKHC6, what you do is, what we saw is that it promotes very efficient, even more than in normal circumstances, adipogenesis. Then, to understand how was this going on, we did the RNA-seq, bulk RNA-seq of all these conditions. And then, first, what we saw is that the treatment with NAD promoted a molecular signature that was unique for all the conditions. And this molecular signature consisted of increased apoptotic processes and diminished translational processes. No? There were a lot of downregulation of transcriptional related processes and upregulation of apoptotic processes, meaning that cells were could not differentiate. They were arrested and they entered into quiescence. The treatment with FKH6 with the inhibitor of SIR1 led to less adipogenic precursors. The cells were arrested in pre-adipocytes. They could not progress into adipocytes, and contrary-wise, the cells treated with FKH6s progressed through adipogenesis and promoted the expression of genes that are related with lipid metabolic processes. No, these genes are very important, like for generating lipid backwalls and all the lipid identity of the cells. We also use this machine that can measure mitochondrial metabolism, and we saw that at day 16, from the progress from progenitors to adipocytes, mature adipocytes, it should come with activation of mitochondrial metabolism through increased oxygen consumption. This is what you see here, no? And a very high increase in oxygen consumption in the cells that are differentiated or treated with the inhibitor of NAD. But the cells that are treated with NAD or the inhibitor of SIR1 do not show this metabolic switch. And then this was important because it also created a link between epigenetic regulation of transcription and metabolism through SIR1 as a key factor. To further explore this system, we use this cell line that is a human embryonic cell line. And we use a CRISPR-Cas system that is a system that has been that was described several years ago and was awarded also the Nobel Prize to these two women, Jennifer Doudna and Emmanuel Carpentier, that allows us to delete genes or to modify the genome with a very high precision that we didn't know before this system was discovered. So we use this system to create SIR1 knockout in a human embryonic stem cell, because this cell line is isolated from the inner cell mass of the blastocyte, and it has the possibility to differentiate to any type of cell in the body. No, it's not the mesenchymal stem cells that you can differentiate to only three lineages. This can be differentiated to any lineage in the of cell, in any cell type. So we generated this SIR1 knockout with a CRISPR, and then we wondered if we could make neurons, for example, no, because the deletion of SIR1 could not make adipocytes, but let's see if it can make neurons. And then we use this this protocol to make mature dopaminergic neurons, and with the SIR1 knockout, what we saw is that they didn't progress this stage. They didn't progress this stage, and the cells were highly heterogeneous. They were not homogeneous, such as in a normal situation. They become other things that we still didn't know until when we were seeing this. And then we wondered what was going on. So to understand this, we also leverage the use of this new technique that has been a huge explosion in the last decade, or maybe less than a decade, that are called single cell RNA-seq, in a way that you can, using microfluidics, you can isolate or identify the transcriptome at a single cell level. It's not like a bulk RNA-seq that you get a bunch of cells and then you do an average of each transcript, how much of each transcript there is. Here you can see the transcriptome at the single cell level, And through bioinformatic analysis, you can identify which kind of cell is in the mixture that you put in the experiment, right? So we did that with the human stem cells, wild type and knockout for SIR1. You can see here in this dimensionality reduction plots. You can every dot is a cell, represents a cell. This is a multidimensionality reduction, these UMAPs, and then here you can see, for example, in human stem cells, you can see that they group together all the wild types and all the knockouts. They group together. No, this in pink. This is the knockouts, in blue the wild types, and this means that they are substantially different between them, and we are still describing these differences between these two groups of cells. And also in neural progenitor cells, we can see that they are substantially distinct. And when we try to give identity to these cells that are in the SIR1 knockouts, what we see is that in the human embryonic stem cells, their identity is all of these cells, almost all of the cells are human embryonic stem cells, as they should be. But in the SIR1 knockout, they become other things, meaning that SIR1 is very, very important to keep the pluripotency identity of human embryonic stem cells. We infer the differential activity of these different transcription factors using advanced bioinformatic analysis. And well, there are several results here that are very important for us, such as, for example, transcription factors that control lipid metabolism, such as RIDP1C, or also some transcription factors such as BMAL that control circadian rhythms are differentially working in these cells. No, we are still integrating all this data because it's a very big amount of data. But also, with the explosion of these techniques, we can now do something that we could not do like 10 years ago, that is the pseudotom analysis. The pseudotom analysis is to define trajectories in the presence, to different trajectories. For example, by analyzing and comparing single-cell RNA-seq profiles, we can theoretically reconstruct a trajectory. That is a path that describes how cells traverse through different states. So you can imagine that you get a bunch of cells that are in different states of differentiation, no? A pre-adipocyte, an adipocyte in day 1, 2, 3, 4, and then you do the single-cell RNA-seq, and then through the bioinformatic analysis, you can assign each one of these cells to a trajectory. You can assign each one of these cells to a trajectory and plot reconstruct or plot the differentiation trajectory based on the value of a pseudotime. That is a number that represents the relative position of a cell in the trajectory, no? And this is a bioinformatic analysis that we can do with the single-cell RNA-seq that we could not do with the bulk RNA-seq. So when we do this in the trajectory of human stem cells to neural progenitors, we can reconstruct perfectly the pseudotime from less differentiated to more differentiated cells, right? Every dot is a different cell, and you can plot it in a pseudotime, and you can infer the trajectory with a lead shot, and it very, very beautifully adjusts to this trajectory. And when we do this with the SIR1 knockout, the trajectory is completely disrupted, it's not there anymore. Meaning that SIR1 is very, very important for differentiation and for neural differentiation in particular. We are leveraging this, just to finish, we are leveraging this research with our collaborator in France through this project that has been recently awarded by the Chan Zuckerberg Initiative, the single-cell neuromed project, to measure metabolism across scales during dopaminergic differentiation in live tissues and organisms, where we want to combine the single-cell RNA-seq techniques or single-cell RNA-seq techniques with maps of metabolism across stages of differentiation using two-photon microscopy that allow us to visualize redox metabolism in live cells and in time and space, no, with subcellular resolution, no. So this is what we are trying to do now in the lab. We are trying to correlate how changes in metabolism and transcription occurs during neural differentiation, and well, I expect to have some nice data soon to show you. And finally, well, these are the lab members and our collaborators and the founding agencies. And well, I want to thank you very much for listening and attending my talk. And I'll be happy to take your questions, if any. Thanks.
Thank you so much, Doctor. Do we have any questions? No questions? Okay. Please remember that if you get any questions further down the line, you can always contact Dr. Lorena throughout her contact information that you can see up in there. And with that being all, we want to give you a little gift on behalf of the committee and our sponsors and the university. Thank you so much for coming and giving us this amazing talk. Can we give her a round of applause, please?
Thank you.
Well, that's it for this talk, guys. Thank you so much for coming. So much for being here. And we will see you back for the science cinema.
Hi.
Because I don't know if I was in one part of the talk.
Uh-huh.
But I wanted to ask if about the experiment you use like any specific speculations with the mice, like they were male, female, like young.
Yeah. No, these mice were well, for this particular research that we did in mice, we include some females, where most of them were mice, male mice, such as, for example, let's say 80% were male mice. But